Good day, my dear postgraduate students. Welcome to the topic on matrix metalloproteinases in neoplastic progression by Gillian Murphy. It is from the recent Advances-22. Please note that this is with relation to neoplastic proliferation or progression. Think of the subheadings. The subheadings will take you to the answer. In this case, the subheadings are quite simple and straightforward, but they should be superimposed on the epithelial mesenchymal transition. This we have already dealt with, and the author also gives it prior to our present topic. This is a simple diagram of the extracellular matrix thanks to Wikipedia. This is an illustration between the extracellular matrix over here in relation to the epithelium, the vascular endothelium, and the intervening connective tissue. So, this is a layer of epithelium. There is a basement membrane. The matrix is over here. Again, the basement membrane of the blood vessel, the vascular endothelium. We shall have it in mind. There are a few synonyms to the topic matrix metalloproteinases called matrix metallopeptidases or matrixins. Usually the topic MMP, matrix metalloproteinases, will be given in the exam. Their function is twofold. One is degradation of the extracellular matrix, and two is the processing of bioactive molecules as well as the calcium containing zinc dependent endopeptidases. So these are the general functions of it. We shall have it in mind. This flow diagram is more important. There are two substances over here, matrix metalloproteinases and TIMP. Inhibitors of matrix metalloproteinases, probably they originate from the tissue, hence the name T. The MMPs, you find that they have a role in inflammation. It is bidirectional. And they also help in the progression of the cancer. Whereas the TIMP, on the contrary, it inhibits the cancer progression. In other words, these two are diametrically opposite each other. We shall be having it in mind. Also, they are activating a few of the mediators promoting the inflammation. The author says that there are about 23 matrix metalloproteinases. But as I was preparing this article, I came across a number of 31. And probably you people will be identifying even more. They are being constantly discovered. 6 of them are anchored to the membrane, and the remaining are present between the surface of the epithelium and the extracellular matrix. So it is a kind of an interaction that is there between the cell surface and the extracellular matrix. Hence, the function shall be degradation of the extracellular matrix, cleavage of the surface receptors, and the release of apoptotic ligands such as FAS. Very simple. I think we should memorize this. Also, what are the general characters of the MMP? You find that they are numerous in number. Already 23 has become 31 or even more. They promote cancer. And there are also non-neoplastic roles of these. They promote numerous non-neoplastic diseases. It is being impeded by TIMP. And the cell behavior is changed in the form of increased proliferation, migration, adhesion, and dispersion. So these are the general characters of matrix metalloproteinases. It was described by Jerome Gross and Charles Lepere in 1962. Apart from the things that have been mentioned over here, there is something called as a cystine switch. So this is diagrammatically depicted, and I took this from the research cave. There are the water molecules externally, And what I am seeing is there is a pro-domain and there is a cell membrane over here. But as these water molecules enter, you find that they get attached to the zinc. Therefore, what was an inactive enzyme gets converted into an active enzyme. Also, you find that there is a displacement of the pro-domain. This is a pro-domain over here which gets displaced. It is getting lost over here. And then there is a degradation of the matrix. And this particular diagram is a relationship between the cell and the extracellular matrix. So this is the cell, I have labeled it. This is the extracellular matrix. And when I say cell, there is a cell membrane. Bimolecular lipid layer. So blue color is the lipid and the white one is the protein. Bimolecular lipid layer between a monomolecular layer of protein. And these will be the various receptors. The labeling is over here, the microfilaments, for example, and then there can be the fibroblast, the elastin, and the collagen, etc. in a matrix. The reference is given over here thanks to Wikipedia. I would like you people to follow this important diagram. I have given a symbol of a key over here. So this is a group of cells, highly dysplastic but somewhat cohesive. And then they are getting changed into a cancer cell. Now, what are the cells that are going to be related to this particular one? There is a macrophage, a neutrophil, a fibroblast, myofibroblast, pericyte, as well as the endothelium, then a lymphocyte. Probably an immunological role is going to be there. After which you find that there is an activation of it. And also you must remember that there are numerous matrix metalloproteinases and there are different numbers for them. And each of them will be acting with a particular cell or site. And some of them can be promoting, some of them can be inhibiting cancer. And also please do observe that there is a TIMP that is given over here. The bad news is time flies. The good news is you are the pilot. Have the exam in mind and keep flying. Michael Alschuler. This is a key diagram in my class. There is a box over here. I would like you people to remember. Whenever there is a complex diagram like this, please break it into sections and then try to analyze them. This is the structure of the matrix metalloproteinases. Look at the colors. Red for signal peptide, green for pro-domain, blue for catalytic domain over here. This is the hinge. And then there is a hemopixin domain, which is yellow. So totally this will be the structure of the MMP. Now what happens is these cells, there are abnormal cells, but then the matrix metalloproteinases can be active for the inflammation, or it can be promoting the tumor group, or angiogenesis, or the breakdown of the extracellular matrix. And for each of this, you find different MMPs have a role to play. And the green color, it indicates a metastatic cell. See here, there is a break in the matrix. It is entering the vascular endothelium, going to a different site, and then it produces a tumor deposit elsewhere, a secondary tumor. So this is a fantastic diagram, which I hope you people will be having in mind. So this is the key to our class. The same thing is given in a small form over here, and I've explained what I told you in the previous slide. The structure of the MMP we have seen. They are usually in a latent form. They are activated by the removal of the pro-domain. We had seen it in the previous diagram. And there is a catalytic domain form of MET-turn. We'll see what MET-turn is. And it can recruit inflammatory cells, promote angiogenesis, as well as degrade the matrix to promote metastasis. So this is what I want. Now the background has been camouflaged and so things are very clear. I find that there is a helical piece of zinc over here, and from which I find that there is a structure of there's MET-turn that is originating. Look at this box over here. This is the MET-turn that is being produced. Probably it is catalytic for this matrix metalloproteinases, which can play a role in the spread of cancer. This is a very important and a very simple diagram. Follow the diagram first and then remember only the text that I'm going to. So these are monomers. A single pair is over here, it's a monomer. And then there is another similar group that is being formed. So it becomes a dimer. And the zinc has got a role to play in it. And these dimers get attached over here. Once they are attached, you find that there is an activation of the TIMP. So inhibitors of metalloproteinases. And this also activates the pro-MMP. See what happens once there is a TIMP, it gets attached. Along with it, it brings the pro-MMP. And once there is an attachment over here, the zinc becomes active and it breaks these. And so what has been attached gets released, probably for activation elsewhere. So I would like you people to kindly remember these headings. There is a monomer that is formed, then there is a dimer, and there is an activation of the dimer over here, and there is a cleavage of the propeptides. Finally, there is an activation followed by the release of the active MMP. These are the steps. I think it can be remembered quite easily. So this is a slide regarding the matrix metalloproteins or transferring proteins in colonic cancer. The proteins that have been mentioned are MMP 9 or 19. So this is a normal picture of a colonic cancer. You find initially there will be an adenoma, then there is a superficial invasion, then there is an invasion across the entire wall. And there are the various metalloproteinases which are involved in each of the steps. These numbers are there. We need not remember them, but only thing we'll have to realize that there are different matrix metalloproteinases for each of the steps. And you find that these matrix metalloproteinases are downgraded in invasion and they are upgraded whenever there is a restriction. And there are different groups which can promote as well as inhibit. And the tissue inhibitors of matrix metalloproteinases, they inhibit the action of MMP. So that is depicted over here. So there is a matrix metalloprotein 9. 9 or 19, they are supposed to be having an important role in the control of it. So this is a tumor. It is an invading tumor, but still I find that they are all cohesive and then a glandular outlet or outline is being well maintained. And they are all positive for the IHC marker. But in this case, I find that though there is a scattered positivity of it, they are very weak. And therefore, what happens is whenever there is a downgradation, you find that or downregulation, there is a weakening of this. So there is an upregulation in this diagram and a downregulation in this, which are opposite, and this can be applied to the Duke's classification. The interaction between the tumor cells and the stromal cells with relation to matrix metalloproteinases is shown in this diagram. We have already seen. You need not remember the numbers for each. So there is a fibroblast, there is a endothelial cell, and then there is the inflammatory cell. There is also a matrix in the middle. You find that this is the cancer cell over here, they have got the receptors. And once they get attached, each of the enzymes are being activated. It can be MMP3 or it can be the vasoexciter growth factor promoting the angiogenesis, vascular endothelial growth factor promoting the angiogenesis, and there can be the MMPs which mobilize the cells or the promote the growth factors. There is ultimately a degradation of the matrix. That is shown over here. So there's a tuft of tumor cells which are about to invade, but then see what is actually happening. There is a substrate over here that is being scissored, and as a result of which these cohesive cells are being broken up into small ones. At the same time, there is the promotion of the angiogenesis. The red lines are the angiogenesis. And finally, you find that there is an invasion of the tumor cell and it can grow at ectopic sites. It's a beautiful article. I would like you people to kindly go to this reference. Life is not measured by the number of breaths we take, but by the moments that take our breath away. Nice quote. So as I had been mentioning earlier, you find that there are numerous types of this matrix metalloproteinases, and they will be digesting various substances such as collagenase, gelatinase, then metalloelastase, etc. And they have been given various numbers. We need not be remembering or memorizing all this. It is only to highlight that so many numbers are present thanks to Wiki Pathways. This shows the categories of matrix metalloproteinases. This is a better classification for us to remember. Based on the number, you find they can be acting on the collagen, gelatin, or the mesenchyme, or some other thing. You find gelatin, fibronectin, laminin, etc. can be acted upon. So stromolysin. So this is one way, and you have got different numbers for it. This is already seen. I will not be repeating it. What are the methods of studying? For that, this actually runs to pages in the book, but I would like you to kindly memorize this particular list alone. There can be a quantitative PCR, RT-PCR, mRNA levels in the matrix metalloproteinases, immunohistochemistry, as I had shown in the previous slide, in situ hybridization for expression of RNA, gene knockout in mice, experimental animals, ELISA, activity assays, and inhibitors also in the models. So these are the various methods for studying the matrix metalloproteinases. Also, a balance between these two will have to be studied, as in this case. So there is a matrix metalloprotein and there is an inhibitor of it. So what is the balance between these two will have to be studied in any one particular case. The entire list is so bewildering, we need not bother about it. It is only to show the number. But the purpose of this slide is to show that there is a role for electrophoresis. This is called as gel electrophoresis, and the substance that is being used is zymograph. So this is a zymographic gel electrophoresis. It can be used to separate the various matrix metalloproteinases. Look at this one. They are all being separated because they are traveling at different speed. Evaluation is different from the modes of diagnosis. What the researchers do is they accumulate the data from the human cancer. They see the various levels of expression at different sites. They identify the enzymes, that means matrix metalloproteinases, correlate with the tissue markers, immunohistochemistry, and the serological level. And they assess the levels of MMP and the TIMP. Animal models are also being used. So they want to decipher a method by which they are able to study the tumor cells so that it can be used for further treatment. It is not for identifying and classifying. In science, in the last 50 years, we had seen physics, but in the next 50 years will be the age of biology. To be frank, this particular topic that we are dealing with belongs to biochemistry. So this is an important one. There are certain substances which are applying a brake. There are certain substances which are promoting the spread. So these are given over here. They can be the inflammation, tumor growth, angiogenesis, etc. The same things are given over here. And there are the various matrix metalloproteinases. Booster, for example, this will be helping in the promotion of it. And these are the various steps: inflammation, tumor growth, angiogenesis, invasion, apoptosis, silenced cancer protective enzymes, matrix remodeling, and the pro and the anti-inflammatory cytokines. So this is a beautiful picture for us to have a clear concept. So this is the matrix over here. In one cell, things are active, and in another cell, it is being impeded. This again shows us the activity of the matrix metalloproteinases in non-neoplastic diseases. So in our answer, we will make a mention of it. CNS, for example, in multiple sclerosis, Alzheimer's disease, cardiovascular atherosclerosis, digestive liver fibrosis, lung emphysema. Others, there can be diabetes, skin diseases, kidney, it can be acute injury or chronic kidney disease, joints, osteoarthritis, rheumatoid arthritis. So, such is the role of matrix metalloproteinases. And to be frank, if you go to the net or to the library, each one of these will be dealt with by an individual article or a research paper. But this is a good slide to summarize the entire thing. So also this. So this is again a beautiful slide. What are all the MMPs which are associated with the various conditions? Neurodegenerative, diabetes, and then gynecological disorders, cardiovascular, etc. And also cancer. So this is the reference of it. It is again a superb one which you people can go to the net and pick it up. It is from the International Journal of Molecular Sciences. MMPs in human disease. The conclusion is MMP is most important. It is the development of technique to analyze the localization of a particular defect. The validation requires a large number of skills. Actually, you people would have visited your thesis. The quantity or the number of the cases required. It has to be fairly large. You cannot be studying just 1 or 2 cases and coming out with a validation. And there is a MMP inhibition theory that is also developing. MMPs are formed by the tumor cells and by the matrix. And TIMP are inhibitors of matrix metalloproteinases. These are the highlights which I have already mentioned also. Looking for validation will prevent you from being yourself. Validation is that every time you want an approval. But if you start looking for that, you will not be able to proceed further. So it will prevent you from being yourself. It will be better that you are yourself and scale the sky. Thank you.